Hi everyone, my name is Alex, and today I'm going to share with you our work on ghost myocytes as undead protectors of the heart. So the heart is one of the body's most essential organs, responsible for pumping blood and oxygen to all of the tissues. Like other organs, the heart needs its own supply of oxygenated blood, which comes from coronary arteries. However, these coronary arteries can be blocked by fatty plaques that build up over time. So this cuts off the oxygen supply of the heart, damaging the heart muscle in what is commonly called a heart attack or myocardial infarction. So these infarcts kill cardiac muscle cells, or cardiomyocytes, which cannot regenerate. But while this may not initially be fatal, these dead muscle cells are gradually replaced by a collagen scar, which stiffens the heart and reduces its ability to pump blood. So this can eventually lead to heart failure, which is one of the leading causes of death worldwide. Currently, it's not well understood how these dead cardiomyocytes are replaced by scar tissue, especially because we can't see these dead cells under a microscope and study them. So we ask the question, how are dead cardiomyocytes replaced by a collagen scar? We conducted our experiments in a mouse model where a coronary artery was tied off to induce an infarct on the heart. And at different time points, the mouse hearts were then removed, frozen, and sectioned onto microscope slides. We then stained these samples with antibodies which bind to specific proteins and fluoresce under a laser. We then imaged these samples with a confocal microscope and analyzed them to measure stain areas. So this is a mouse heart after an infarct. And along here is the wall of the left ventricle, which is made up of cardiomyocytes. Now, naturally, viable cardiomyocytes have a protein called troponin I, which is shown here in cyan that allows them to contract. These dead cardiomyocytes, they lose their troponin through membrane permeabilization. So we have this distinction between the alive cardiomyocytes in bright cyan, and then the dead cardiomyocytes are dark and invisible, like throughout the infarct region here. But these dead cells are permeable from holes in their membrane, which means that proteins like troponin can escape, but also chemical dyes like Evans blue can enter these dead cells. So we injected Evans blue one day after the infarct. And in yellow here, we can see that it highlights all of the dead cells in the infarct region. And if we look even closer, we can tell that these dead cells have maintained their cardiomyocyte structure, despite losing functional components such as troponin. So thus, we've termed them ghost myocytes. And now that we can see the ghosts, we can study them. So what is the fate of these ghost myocytes? At day 3, we have an infarct here, and you can see that there's many of these yellow ghost myocytes all throughout the region. But by day 7, many of these ghosts have vanished. And yet, as you can see, we have much less yellow Evans blue signal. So something's clearing the ghost myocytes. But does this clearance have a relationship with scar formation? Shown here in magenta is collagen 1, which is a critical component of a scar. And at day 7, coinciding with the increased breakdown of ghost myocytes, we have an increase in collagen scar formation. But where exactly is this collagen being deposited? So if we take a closer look into the infarct at day 3, we can tell that the collagen in magenta surrounds the yellow ghost myocyte region. By day 7, when this yellow footprint has shrunk, the collagen has taken over these cleared areas. So the ghost myocytes, they seem to act as a barrier and hold back scar formation. And as they are removed, the scar expands, further stiffening the heart and reducing its ability to pump blood. So we wondered what's actually clearing these ghost myocytes. And from other work in our lab, we know that the immune cells respond to the heart during an injury. And by staining for macrophages, we can see that macrophage recruitment again follows this pattern of surrounding the yellow ghost myocyte footprint. And based on their role in clearing debris, we hypothesized that macrophages are responsible for clearing the ghost myocytes. So future experiments will attempt to confirm this by inhibiting macrophage recruitment and seeing the effects on clearance. So, in conclusion, we've addressed key components of how dead cardiomyocytes are replaced by a collagen scar. So we found that after an infarct, dead cardiomyocytes lose their troponin while maintaining their overall structure. We've also found that we can label these newly termed ghost myocytes with a dye called Evans blue. And by using this new technique to visualize dead cells, we've seen that by day 7, a lot of these ghost myocytes are cleared, and their clearance allows a collagen scar to form. But until they're cleared, the ghost myocytes seem to occupy space and hold back scar formation, almost protecting the heart from beyond the grave. Thank you to all the people in the COOPS, Deniset, and FEDEC labs who have helped with this project. I'd like to thank Brian in particular for his mentorship on this project as a subset of his larger work. Thank you for listening.